Thalassemia is a genetic disorder that runs down in families. Basically, it is due to it results in the production of abnormal red blood cells that are destroyed during the early period of their production. And because of this, it leads to the deficiency of red blood cells in the body and cause anemia. That's the reason this thalassemia is known as one of the type of anemia, and it is classified as the type of anemia. In our today's lecture, we are going to study about the pathology of thalassemia. We will see that how thalassemia is further classified, and we will also focus upon the pathogenesis of the thalassemia, how it progresses. So let's start our lecture. As I told you that thalassemia is a disorder that results in the production of abnormal red blood cells. So basically, the production of these abnormal red blood cells is due to the defects in the globin chains that are part of the hemoglobin. Hemoglobin is the important part of our red blood cells and that it's a protein that makes the major portion of the red blood cell and carry carbon dioxide and oxygen inside our body. So obviously when there is a deficiency of hemoglobin or there is the presence of abnormal hemoglobin, there will be the production of abnormal red blood cells and that will be destroyed during the early period of their production. There will be deficiency of the red blood cells in our body. So the oxygen carrying capacity of the person will be decreased and also some other functions will be affected that I will tell you when we will discuss about the clinical features of the thalassemia. So before going into all of these things, we will discuss what is the types of the hemoglobin that can be present inside our red blood cells. So basically, there are different types of hemoglobin that can be present inside our red blood cell, out of which some are normal, some are abnormal. Basically, the normal type of the hemoglobin that is present in a normal adult is the type A. The hemoglobin A is present that consists of the 2 alpha and 2 beta chains. Now let me tell you here that hemoglobin is composed of 4 chains. 2 alpha chains and 2 non-alpha chains. Now these non-alpha chains can be beta chains, can be gamma, delta or the zeta chains depending upon the condition of the person, whether he is suffering from thalassemia or he is a normal individual, infant or so on. So in a normal adult, there is the 2 alpha chains and the 2 beta chains, and that the hemoglobin type is the type A hemoglobin. Then there is another type of the hemoglobin that is the A2, the type A2 hemoglobin. This type of the hemoglobin consists of 2 alpha chains and the 2 delta chains. Basically, this delta chain is present when there is during the early childhood, before the age of 6 months, this delta chain can be seen in the childs. Then there is another type of hemoglobin that is the F hemoglobin. Hemoglobin F basically consists of 2 alpha chains and 2 gamma chains. Now this gamma chain is present during the intrauterine life, and after that this is replaced by the beta chain or any other chain depending upon the genetic information of that person. So in case of the beta thalassemia, mostly in case of the beta thalassemia where there is a deficiency of the beta chains, this type of the globin chain can be seen, and it may be the part of the hemoglobin that results in the beta thalassemia. Then there is another type of the hemoglobin that is the hemoglobin H. Now this type of the hemoglobin consists of 4 beta chains only. Now that means that person is deficient in alpha chains. So we can say that person is suffering from the alpha thalassemia, where there is a deficiency of alpha chains in the body. So whenever there will be the alpha thalassemia, then the type of the hemoglobin that will be produced inside the inside those person will be the type H hemoglobin. Then there is another type known as the Bart's hemoglobin. Bart's hemoglobin consists of four gamma chains. Now there is no alpha and no beta chains. As I told you that this type of the hemoglobin chain is only present during the intrauterine life. That's why when the baby is born, he will suffer from the severe symptoms of the thalassemia, and usually it is very difficult for the baby to survive. Then there is another type of the hemoglobin that is known as the Portland hemoglobin, and it consists of two gamma chains and two zeta chains. Now, zeta chain is basically present during the early intrauterine life, during the early development of the fetus, and then it is replaced by the gamma chain and beta chain, so on. So, whenever there will be any defects, there can be the presence of both these type of the chain as well. That will be called as the Portland chain. So these are some types of the hemoglobin that are present either in normal person or the person suffering from the thalassemia. And also by knowing what type of the chain is present, hemoglobin chain is present inside the person, we can also distinguish the type of the thalassemia a person is suffering from, and then can treat accordingly. So moving on, now you guys know about the hemoglobin, you know about the genes and structure of the hemoglobin and its various types. Now let's move on to the molecular pathogenesis of the thalassemia. Now, as I told you that it is a genetic disorder. It is a genetic disorder. That means that there is some defect in the genes that are present on the chromosome. So obviously there will be some defects in the genes that are present in the chromosomes, and the chromosomes that are involved here in thalassemia are the chromosome 16 and the chromosome 11. So basically these are the two chromosomes that consist of the genes encoding for the hemoglobin chains. Different types of the hemoglobin chains. Like on the chromosome 16, there is the genes are present that encode for the alpha chains, and genes are present that encode for the zeta chains. Now these are the globin chains. You know that these are the globin chains: alpha, beta, gamma, zeta, and delta. Now these genes then further result in the formation of the different chains, the different combinations of the chain, and they are basically encoding for the formation of hemoglobin chains. Then on the chromosome 11, there is the presence of gamma genes that encode for the gamma chain, delta chain, and the beta chain, and they further lead to the formation of different type of the hemoglobin. That is hemoglobin A, that consists of the beta chain, two alpha and the two beta chains, and then the A2, that consists of the two alpha chains and the two delta chain. Then hemoglobin F, that consists of the gamma chain along with the alpha chain, or maybe they consist of the zeta chain as well, like in case of the Portland hemoglobin. Then there is some embryonic type of the hemoglobin that consists of the zeta chain and the type of the globin chains in it. So basically these are the two genes, the two chromosomes that will consist of these genes encoding for the different type of the globin chain. Whenever there will be some inherited defect in these genes, that will lead to the alteration in the structure of hemoglobin, and that will lead to the defect called as the thalassemia. Now, before telling you about the beta thalassemia, let me tell you that there are different types of the thalassemia. That is, mostly there are the two main types, the alpha and the beta thalassemia. As we have said that in a normal adult, in a normal individual, there are two types of the globin chain, two alpha chains and the two beta chains. That means that four chains are present in the hemoglobin. So whenever there is a deficiency of the beta chains, either the beta chains are completely absent or they are reduced, they're deficient, then the person will suffer from the beta thalassemia. As the name indicates, there will be the deficiency of the beta chains in the structure, and as it is a genetic disorder, then this obviously this will be inherited. Now beta, there will be some defect in the chain, in the gene that is encoding for the beta chain, and this defect can be present in one parent, or can be present in both parents, or can only maybe present in the recessive form in one parent, and then it is further transferred onto the children. So one or more than one children may be suffering from the beta thalassemia. So basically the beta chains will be replaced by another type of the globin chain that will result in the formation of the beta thalassemia, and it is the most prevalent type of the thalassemia in the patients. Now, what is the molecular pathogenesis of the beta thalassemia? You know that beta thalassemia, the gene that will encode for the beta thalassemia, is present on the chromosome 11. So obviously there will be the defect is involving the gene, and the chromosome 11 will be involved in the beta thalassemia. Whenever there is a beta thalassemia, there will be either the complete absence or reduction in the beta chains, beta globin chains. That will, in other words, that will lead to the excess free alpha chains. Now, the excess free alpha chains may be due to the reduced level of beta globin chains, or due to the absence of beta globin chains completely. When these alpha chains are in excess, what will happen? They will cause the membrane damage to the peripheral red blood cells, because the hemoglobin is a part of the red blood cells, and that's why they will interfere with the membrane of the red blood cell and will cause the damage to the red blood cells and will lead to the hemolysis. Now, this is the thing that I told you during the start of the lecture, that these red blood cells are destroyed during the early period of their production, and that is due to the excess of the alpha chain that will then interfere with the membrane of the red blood cells and will cause the destructions of the cell before they leave the bone marrow and enter the blood circulation. Then there can be another case. These excess free alpha chain may lead to the precipitation in RBC's precursors within the bone marrow that lead to the premature death of RBC's precursor. Now, the precursor cells, the stem cells, they can also be affected by these excess free alpha chains that may lead to the premature death of the precursors. As there is the precursor cells that have to be converted into the erythrocyte, to the red blood cells, they are not present or they are deficient, obviously the process of erythropoiesis will be affected or reduced. Both of these things, either the hemolysis is occurring or either there is ineffective erythropoiesis, what will be the end results of both of these things? The person will suffer from the anemia. Obviously, there will be the deficiency of the red blood cells, there will be the deficiency of the hemoglobin, so the person will definitely be anemic. Now, moving on, what will happen? There will be either the as a result of the anemia. Now, you know that when there is a decreased level of red blood cell production, there is a signal is sent to our kidney that release erythropoietin. So in this case, what will happen? Either the production of erythropoietin will be increased, or there will be increased absorption of the iron. Body may get like there is insufficient iron. That's why the globin chains, the beta globin chains, are not being synthesized in the body. So more and more iron will be absorbed. Or there will be the need of blood transfusion in severe cases that will also have the effect. Like when there will be the increased iron when there will be the increased erythropoietin, that will lead to the bone marrow expansion, as it will tend to produce more and more red blood cells that will cause these skeletal deformities, that will cause the skeletal changes and hyper metabolic state, and there will be some skeletal defects, structural defect in the body due to this condition. Then, because of the excess absorption of the iron and because of the blood transfusion, as the blood that is transfused to the body, it also consists of the iron. So both of these things may lead to the iron loading. There will be an increase in the level of the iron inside the body that will lead to the different conditions, like it may lead to, or it may be a risk factor for the liver cirrhosis, for the endocrine dysfunction, and for it may be damaging for the heart, as it may lead to the arrhythmia, or it can also lead to the heart failure. That's why it will cause different symptoms in the body that may be related to the liver, the heart conditions, the pulmonary symptoms, as there will be the decreased oxygen transport as well, and some symptoms that are related to the endocrine dysfunction. So basically, this is how beta thalassemia is going to affect our body. Now, there is the production of abnormal red blood cell. And we know that in the beta thalassemia, there is the production of abnormal red blood cell. So let's see that how these red blood cells will look like. This is the diagram. This is a microscopic slide that is showing the cells affected from the beta thalassemia. In the case of the beta thalassemia, the red blood cells that are produced, they will be hypo-chromic, and there will be the microcytic, and also the shape of these red blood cells will be destroyed, or will be different from the normal red blood cells. So basically, these characteristics will make them different from the normal red blood cells. Now, the pathology of the thalassemia will be different in case of the homozygous condition and in case of the heterozygous condition. The case when the beta thalassemia is homozygous, or the carrier is homozygous, then the symptoms will be more severe, and there will be the more severe microcytosis in the patient who is suffering from the homozygous type of the beta thalassemia. There will be more microcytosis. There will be the patient will be hypochromic, like that is a general characteristic of the thalassemia. And there will be the shape will also change more markedly, and there will be these basophilic stapling. That means that there will be the presence of numerous basophilic granules inside the that you can see in the slide, and also there will be the presence of other cells in case of the heterozygous condition. Now, in case of the heterozygous condition, there will be the microcytosis, there will be the hypochromic condition, but all of these things will be less seen, or it can be seen less as compared to the homozygous condition. In case of the heterozygous condition, there will be the heterozygous carrier, and there will be the microcytic anemia, hypochromic condition, but that will be less apparent as compared to the homozygous condition. And also there will be the less amount of the basophilic cells seen as compared to the homozygous condition. Rather, here more reticulocyte will be seen as compared to the homozygous condition, and the shape of the cell that will be somehow more relevant to the normal red blood cell as compared to the homozygous condition. So we can say that there will be the more changes in case of the person who is suffering from the homozygous condition, and less severe changes in the person who is suffering from the heterozygous condition. So you can also distinguish this from the microscopic study that the patient is suffering from the homozygous or heterozygous condition, as these things are making different and making both of conditions different from each other from the microscopic point of view. Now, moving on to the clinical symptoms or the clinical features of the beta thalassemia. We have studied the pathogenesis of the beta thalassemia, and when we were discussing the pathogenesis, we also discussed that how it will lead to anemia and how or what type of the symptoms it can produce. So let's get into the more details of the clinical features of the beta thalassemia. We know that there is a decrease in production of beta genes, or there is the lack of the beta genes during the beta thalassemia. There can be the three consequences of it. Either there can be the relative excess of production of alpha genes, means all of the genes that are being produced, they may be the alpha genes, or that in case of the homozygous beta thalassemia, there can be the formation of the tetramers, the alpha tetramers, that can lead to the formation of the Heinz body inside the red blood cells. That is also a characteristic of a type of the thalassemia. So there can be a condition where there is the relative excess production of alpha genes, or there can be the relative excess production of the gamma genes. That means there is the presence of alpha genes along with the gamma genes, or there can be the HbF type of the hemoglobin present that consists of RBCs that show selective survival. Now, in the case when there is the presence of gamma chain along with alpha chain, and there is the absence of the beta chains, there will be the increased amount of the HbA2. As the HbA2 is a type of the hemoglobin, you know that where there is the presence of two alpha chains and the two gamma chains, that is known as the A2 type of the hemoglobin. So there may be the increased level of this type of the hemoglobin that may be a cause, and in case where there is an excess of alpha chains, this may increase the instability of alpha chains tetramer. As I told you that there may be the presence of 4 alpha chains, that is the formation of a tetramer that will lead to the unstable chain formation. Tetramers will precipitate either in the bone marrow or the outside the bone marrow. What will happen inside the bone marrow? Inside the bone marrow, there will be the ineffective erythropoiesis, and outside the bone marrow, there will be the hemolysis. Like we have talked about this, and both of these things will lead to the anemia. And what will be the consequences of the anemia? If we talk about the treatment that will be carried out, or the anemia, the way we treat it from the exogenous source is that there will be the increased intake of the iron or the absorption of the iron, or there will be the blood transfusion that will lead to the increase of the body iron, or endogenously there will be the increased erythropoietin that will lead to the extramedullary hematopoiesis or increased intramedullary hematopoiesis. Then what will happen now in case of the production of HbF? In this case, when hemoglobin F is being produced, what will happen that there will be increased in the affinity of the hemoglobin for oxygen. Now, the affinity of oxygen binding with the hemoglobin is increased, so there will be the more binding of the oxygen to the hemoglobin. But this hemoglobin can bind to the oxygen, but it is unable to transport this oxygen to the tissues, or to transfer this oxygen to the tissues. So what will happen in this scenario? There will be the increased affinity of hemoglobin for oxygen, but there will be the decreased tissue oxygenation. In this case, what will happen that tissues will send signal to the brain that there is a deficiency of oxygen, maybe there is the deficiency of the hemoglobin and the red blood cell. That's why the brain will send signals to the kidney, and the kidney will release the erythropoietin. There will be an increased release of erythropoietin that will then ultimately start the formation of erythrocyte. That is, it will increase the erythropoiesis process, but obviously when there is a defect in the genes, the defect in the formation of the protein, hemoglobin, that will that's why the deficiency could not be compensated, and there will be increase in both intramedullary and extramedullary hematopoiesis, that is the formation of red blood cell inside the bone marrow and outside the bone marrow, some other places inside the body as well. So summarizing all of these things, the beta thalassemia can lead to anemia, and it may manifest as the iron overload, the symptoms that are related to the iron toxicity, the symptoms that are related to the liver, that are related to our heart, like arrhythmia, like tachycardia, and the symptoms that are related to our pulmonary symptoms, and also some skeletal deformities. Telling you more about this, there are two types of the thalassemia. The beta thalassemia, it can be the homozygous, or it can be the heterozygous. Homozygous type of the beta thalassemia is more severe as compared to the heterozygous beta thalassemia. Heterozygous beta thalassemia is less severe and cause minor symptoms as compared to the homozygous form, and it is also called as the minor thalassemia minor. Now, what is beta thalassemia major and minor? The thalassemia can be classified into two types further, the major and the minor one. Thalassemia major is the form of the thalassemia that is more severe and cause more intensity of the symptoms as compared to the minor, that is a mild form of the thalassemia. Now, how you can distinguish apart from this difference between the major and the minor thalassemia? Major thalassemia, in case of major beta thalassemia, there will be the hypo-chromic anemia, there will be the severe red cell dysplasia, the HbA hemoglobin, that is a normal hemoglobin that can be present in an adult, its level will be decreased. Instead, the level of the HbF will increase. And we have just studied that when this level increases, the tissue oxygenation will decrease, and there will be the production of the binding of oxygen will only increase. That will lead to the increase in the erythropoietin hormone and erythropoiesis. Or there will be the erythroblastosis, the formation of abnormal red blood cells, and also there is the family history in case of prominent family history in case of the thalassemia major. In case of the thalassemia minor, there will be the mild anemia as compared to the thalassemia major, and there will be the microcytic hypo-chromic red blood cells that will be produced, like in case of the major thalassemia. Then there will be increased resistance to osmotic lysis. That's why it will cause minor symptoms as compared to the major form. There will be increased HbA2, the type of the hemoglobin that in which there is the presence of the gamma chains instead of the HbF. There will be the formation of HbA2, and there will be the punctate basophilia, and also here there will obviously there will be some family history that will lead to the cause of the disease. So these are the two types of the thalassemia, major and the minor. The one with the more severe disease and the one with the less severe disease and symptom. In case of the major, there will be the severe red cell dysplasia, and the type of the hemoglobin that will be present is HbF. Whereas in case of the minor, the type of the hemoglobin that will be present is HbA2. Now we have discussed a lot about the beta thalassemia, and let's talk about the another type of thalassemia, that is the alpha thalassemia. Now, what is alpha thalassemia? Alpha thalassemia is basically when there is a deficiency in alpha genes. You know that normal hemoglobin is composed of alpha and the beta genes or non-alpha genes. Whenever there is the deficiency or decrease in the alpha chain, there will be the amount of the other non-alpha chains will increase, and this will lead to the alpha thalassemia. Now again, this condition can be homozygous or heterozygous. Maybe there are 4 beta chains present, maybe along with the beta chains some other chains are present, that will then obviously lead to the manifestation according to the type of the hemoglobin that is present inside our body. Now, depending upon the type of the defect, whether there is a defect on the single alpha gene or both alpha genes, where there are 3 or 4 alpha genes inside the body, there will be the manifestations of the symptoms. Depending upon these things, the disease can be the homozygous, heterozygous, or it can be the patient may be a silent carrier. So all of these things will define the type of the disease that will be inherited from parent to the child. The patient may be symptomatic, may be recessive carrier, or may be asymptomatic as well. That we will discuss in our coming slides, when we will talk about the different types of the alpha thalassemia. So let's talk about the molecular pathogenesis of alpha thalassemia. Now, in normal cases, what happened that in normal cases, you know that chromosome 16 is responsible for this defect, because the genes for the alpha chain are present on the chromosome 16. Now, in normal case, what happened that on the 2 chromosomes there are the 4 genes, and all of the 4 genes are present in case of the normal case. But in case of alpha thalassemia, what happens that there is defective genes that may be present. Either mostly in most cases there is a deletion of the gene, but there can be the presence of defective gene as well, or the mutation can be there. So whenever there can be the cases when only one gene is deleted from one chromosome and one is deleted from another chromosome, or there can be a condition when 2 genes are deleted from the same chromosome, or there may be a condition when only one gene is deleted from the one chromosome. So depending upon these characteristics, the symptoms and the appearance and characteristics of alpha thalassemias are different, and that also tend to classify the alpha thalassemia into its further traits. So basically, it will due to the defects on the chromosome 16. Now, some patients are silent carrier of the alpha thalassemia. As I told you, that when there may be some inheritance where the person may be recessive, and the sign and symptoms does not appear, and simply he acts as a vector, and simply transfer the disease to next generation. In case of alpha thalassemia, the patient is asymptomatic only when there is only when there is a deletion of one gene on one chromosome. Now, we know that in normal case there is a presence of 4 genes, but when there is the deletion of one gene, what will happen that the patient will be asymptomatic, and the type of alpha thalassemia will be called as the silent alpha thalassemia, or the patient will be the silent carrier of the alpha thalassemia. As this type of patient is asymptomatic, so it is mostly this type of the alpha thalassemia is not diagnosed, except in case of diagnosis of other diseases, or during the genetic testing, it may present that the patient is suffering from the alpha thalassemia. But usually the patient go undiagnosed and do not present any symptoms. Then there is the hemoglobin H disease. Now, what happens in hemoglobin H disease? You have studied about the type of the hemoglobin H. Obviously, that will be more prevalent in case of this type of the thalassemia. Now, in case of this type of the thalassemia, what will happen that there will be the deletion of the 3 genes out of the 4 genes. So you can say that it is the severe type of the alpha thalassemia that will lead to the formation of the Hin's body as well. Here what will happen that 2 genes from the same chromosome, one chromosome and one from another chromosome will be deleted. So total 3 or 4 genes are deleted that lead to the causing a deficiency of alpha globin chains and leading to the alpha thalassemia. Now, this hemoglobin H disease will may manifest as the microcytic anemia, and it may lead to increase in hemoglobin Bart level, or there will be the variable levels of hemoglobin H. And also there will be the formation of the Hin's body. Now, depending upon the type of the chain that is replaced by the alpha chains, or there may be the presence of H type of hemoglobin or hemoglobin Barts as well. And what is these Hin's bodies? These Hin's bodies are basically small inclusions that are present at the mid of the red blood cells, and these are the accumulation of the abnormal type of the hemoglobin that is formed inside the red blood cells. So they are called as the Hin's bodies. Now, homozygous type of the alpha thalassemia. In homozygous type of the alpha thalassemia, there is the marked anisophylocytosis. That is, the shape of the red blood cells will be highly destroyed, and it will be much different from the normal red blood cell shape. And also there will be the presence of HB Bart. There may be the heart failure. The patient may suffer from the heart failure. The patient may suffer from the generalized edema, hepatosplenomegaly, as spleen is involved in destroying the abnormal type of the red blood cells, and liver. There is the synthesis of the red blood cell when there is the extra medullary synthesis of the red blood cells. Then there is the obstetric complications that can also arise due to this type of the thalassemia. Now, like when we were studying about the beta thalassemia, this homozygous alpha thalassemia is much more severe as compared to the heterozygous form of alpha thalassemia. This type of thalassemia will lead to more severe symptoms, as you have seen, like the heart failure, obstetric complications, and hepatosplenomegaly, as compared to the heterozygous type of the thalassemia, as there can be the removal of genes from both of the chromosomes. So this was our today's lecture, where we studied about the thalassemia as a type of the anemia, and we have discussed that how thalassemia lead to anemia. So summarizing what we have studied about the thalassemia in our today's lecture, we started with the definition of the thalassemia and classified into 2 types, the alpha and the beta thalassemia. Then we talked about the hemoglobin, the different type of the hemoglobin, talking about the normal hemoglobin, the one that is present in the alpha thalassemia, beta thalassemia, the one that is present in the infants, and the one that is present during the intrauterine life. We talked about the hemoglobin A, A2, H, hemoglobin Barts, hemoglobin F, and we talked about the Portland hemoglobin as well. Then moving on, we discussed in detail the beta thalassemia, that there is a deficiency of the beta chain and excess of the alpha chain. that disrupt the membrane of the red blood cells, and that will cause the hemolysis, or in other perspective, it may lead to the destruction of the precursors of the red blood cells, that will lead to the ineffective hematopoiesis, effective erythropoiesis, and that will lead to the anemia, the deficiency of red blood cells, and ultimately there what will happen, that there will be the iron overload, increased amount of the erythropoietin being produced, and all of these, or the blood transfusion, all of these things will increase the iron overload on the body. There will be the extra or the erythropoiesis will also increase. So there will be the skeletal deformities, there will be other signs and symptoms that will be produced due to the iron overload. Then we talked about the pathology of the beta thalassemia, and we discussed that how cell shapes and there can be the microcytic or macrocytic anemia, and hypo-chromic cells are being produced during the beta thalassemia. And we also discussed about the homozygous and heterozygous type of the thalassemia. Then we discussed about the major and minor thalassemia, minor and major, and we distinguished them as thalassemia major is more severe and thalassemia minor is less severe. Also, the major one consists of F hemoglobin, and the second one, the minor one, consists of HbA2 hemoglobin. Then we discussed about the alpha thalassemia, and we discussed that it is due to the defects in the gene present on the chromosome 16. There can be the defect in the only one gene, or there can be the defect in the 2, 3, and the 4 gene as well. And depending upon this, the patient's symptom and disease severity will change. The patient may be the silent carrier. There can be the H disease, and there can be the homozygous condition that lead to the more severe symptoms as compared to the other forms of the disease. So that's all that we have discussed in our today's lecture. Hope you guys got enough about the thalassemia. Thanks for watching sqadia.com.